Chapter 5 will discuss the protein purification and the characterization technique, all right? So chapter 5, so first of all, the first topic is to discuss the extracting the pure protein from cells and the color chromatography. So today, I hope that we just finish these two topics, all right? To extract the pure protein from cells and then to perform the color chromatography. So there are two different systems that you can grow the microorganism, because to extract the pure protein from cells, they say that commonly used organism will be the bacteria cell, right? The microorganism. So you need to grow the bacteria cell in the media, right? And then you have two different type of the growth condition. It can be a batch culture, like what normally people do in the laboratory. You prepare the media and then put the media in the flask, and then inoculate the cell, inoculate bacteria cell, and then let the bacteria cell grow in the flask, all right? And then after they saturated, then you perform the centrifugation to collect the pellet of the cells. And then from these cell pellets, then you break the cell and then to release the cell extract and then to isolate the protein. And again, for the continuous culture at the industrial level, so you have this fermentation and then try to continuously to grow the bacteria cell, all right? In the meantime, you continue to add the fresh media, all right? And so that you can grow to you can maximize the production of the protein, okay? And then of course the protein are valuable, complex, and fragile product, okay? And then there are two major phases that can be used in producing the protein. So the one with upstream, so you express the protein, you grow the cell, express the protein, and then you prepare the cell extract. And downstream, there'll be purification of the protein, okay? And then to isolate the protein from cell, okay? You have many steps needed to extract the protein from the cell and to separate from the contaminant. And before the purification begins, right, those proteins must be released from cells through the homogenization. So that means you disrupt the cell to let the cell, you break the cell to to release the component, and that including the protein of interest. And this process we call homogenization, okay? And this is just the example of a protein purification scheme to show you the amount of from the crude extract, all right, and the total protein. And then if you go through the salt precipitation, and then the protein number, and then ion exchange chromatography, and this molecular sieve, which is size exclusion chromatography, all right, and to show the recovery from 100% for the salt precipitation, you get 48% ion exchange, that's the 29%, size exclusion, 23% recovery, and this is the immunoaffinity chromatography, purification 11% of the recovery, okay? So that's just to give you some overview of the different technique, the recovery rate from the different technique to recover the protein. And then the salt precipitate, right, to sort out. So because after protein solubilize, they can purify based on the solubility, all right? And depending on the overall charge, ionic strength, polarity. And then the ammonium sulfate, okay, these ammonium sulfate usually are used to sort out the protein, okay? But before that, you need to perform the differential centrifugation, right? Differential centrifugation, so that you can remove the contaminant, right? So you can separate the cell component. And then to give you a more pure pure extracts and easier for you to isolate the protein. Okay, and then one of the commonly used technique to isolate the pure protein is a column chromatography. All right, column chromatography, there are two different phases. We have stationary phase and mobile phase. The stationary phase, okay, that's a sample interact with this phase. That's the resin. That's the so-called resin that we use to interact with our sample. The mobile phase, basically, that contains the sample, the protein of interest. So you have to run the mobile phase through the stationary phase, and then through the interaction from the protein of interest with the resin, then you will be able to isolate the pure protein. And this is a typical, just the typical example. So you have a column, and then you have resin, all right? And then the sample goes through the column, and over the time, then because the property of the resin, they can separate the protein based on the specific property, okay? And then those protein, you can separate different things through the column, and finally you can elute the specific protein of interest you want, okay? And then the first one I'm going to discuss with you is the size exclusion, which is gel filtration chromatography. So this column will separate the molecule based on the size, based on the molecular weight of the protein. So the stationary phase composed of cross-linked gel particle, all right? And this gel particle can determine the cutoff of the molecular weight that you want to purify, okay? Because they control the pore size, and the molecule smaller than the cutoff molecular weight, and they will enter the pore, so they will be trapped by the resin. And the large molecule, those molecules greater than the cutoff molecular weight, will not enter the pore, so they will be excluded from the resin. Therefore, they will be elute out faster. So just like this, so we have this size exclusion column, and then you run the sample through the column. So this, for example, this green molecule will be the large protein, and the pink molecule will be small molecule. So those large molecule will run through the column faster. Therefore, you will collect those protein first, and then later on, those smaller protein will also eventually come out of the resin. Then you will be able to collect it. So that's how you separate this protein based on their molecular weight, and so we call this a size exclusion chromatography. So here it's more detailed that because those resin, they have this pore, so the smaller molecule will enter the pore, the sieve, and large molecule be excluded. And so that's why these large molecule will go through the column faster than the smaller molecule. Okay, so that's gel filtration separated by the size. And then affinity, the affinity chromatography. All right. So basically, you use a specific binding properties of molecule protein. All right. Use the stationary phase has a polymer that can be covalently linked to a compound called a ligand, and that specifically bind to the protein of interest. Okay. So basically, the principle of affinity chromatography, that's in a mixture of a protein, only one bind to the substrate. One bind to the substance, that's substance S, that's a substrate, okay? And the substrate is attached to the column matrix. Once the other protein have been washed out, then the protein who bind to the P1 that bind to the S, then can be eluted, all right? You just use the high concentration of the S, because that will perform the competitive binding. That's how you elute the P1 here. Okay, that's affinity chromatography. And this is just to show you the group-specific affinity resin. Okay, the resin that people use, and the ion exchange. So ion exchange chromatography, and basically that's interaction based on the overall charge. Okay, it's less specific than the affinity because the affinity chromatography will be very specific. You can target to a specific protein if you use the antibody to purify the protein. So you can just target to very specific protein. For ion exchange, based on the interaction of overall charge. Okay. So in this case, we have this cation exchange beads, right? So cation exchange bead, I mean, they will bind a positive charge, the amino acid, okay? And then for the anion exchange beads, they will bind the negative charge the protein. And then so this is the example sort of ion exchange. So when you go through the protein, so you have this ion exchange resin, all right? So this one, they will bind the negative charge.
Na?
Yeah, so this one is this is cation exchanger. So they will cation exchanger. So basically, the cation exchanger, because these resin are negative, they bind a positive charged amino acid. All right, this is cation exchanger. So those sodium, all right, and the negative charge here. So basically, they will bind those positive side chain. All right, and then those negative side chain that will go through fast, and later on those positive charged side chain, they can be eluted. So this is the example of a cation exchanger. So at the beginning of the separation, various proteins are applied to the carbon. Then the carbon resin is bound to the sodium counterion. Okay, the sodium counterion, all right? And then the protein, in step 2, the protein that have no net charge or a negative charge will pass through, all right? If no net charge or negative charge, they will pass through these quickly, all right? And then it will come down to the bottom. And protein that have a net positive charge will stick to the carbon, right? Just a second, Hadil. And then now displacing. So they will bind and displacing those sodium ion, all right? They will displace the sodium ion. And then at step 3, all right, excess of sodium ion is then added to the column. Then step 4, the sodium ion outperform the bound protein for the binding side of the resin. Then the protein is eluted. Okay, so this is the example of the cation exchanger. Okay, Hadil.
So the one that went down in the beaker in step 2, it looks like it has 3 minuses, right?
So in the step 2 here, basically, so remember this is a cation exchanger. So the protein without charge or the negative charge will run through these resins directly to the bottom. And those protein with overall positive charge, then they will displace those sodium ion and they will bind to the resin. Okay, and then later on, if you use more sodium, all right, so you will be able to elute those positive charged protein. Okay, so this is ion exchange chromatography and example of a cation exchange. And this slide here just to show originally see the positive charge, then you have negative charge group, and also this is anion exchange, all right, anion exchange. So you have low concentration, and then those are negative charge protein. They will bind, all right, they will displace those original negative charge on the resin, and then bind here. And later on, increase the salt concentration. Then you can elute those negative charge protein. So this is an anion exchanger. And then the hydrophobic interaction, chromatography. So basically, the protein are salty on basis of their repulsion of water. All right, so the column beads coated with the hydrophobic molecule, and then the hydrophobic amino acid in the protein will be attracted to those beads. And later on, you reduce the salt concentration, then you will be able to elute the protein. And the resin, basically, these two types of resin commonly used, the phenyl and octyl substitute resin, because they can interact with non-polar amino acid side chain protein. And protein released by lowering the salt concentration. When you use the low salt concentration, then you can elute the protein. And this is just example to show you that originally you use the high salt buffer and to make those hydrophobic side chain to stick out, so they will interact with the hydrophobic ligand. All right, and later on, when you reduce the salt concentration, then you can elute this protein. So this is the hydrophobic interaction. Okay, and now I just want to show you a video about the affinity chromatography. Okay, the affinity chromatography. This is a principle.
Affinity chromatography depends upon the reversible absorption of biomolecules through biospecific interactions on the ligand. Most affinity chromatography experiments are performed in three main stages: equilibration, sample application and wash, and dilution. The first step is the equilibration of the stationary phase to the desired start conditions. The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out all unbound material. Non-binding molecules will pass through in the flow-through during washing with binding buffer. In the third step, elution, biomolecules are released from the biospecific ligand into the elution buffer by change in the buffer composition. A common way is to increase pH of the buffer.
So that's the example of the affinity chromatography. Then I'll show you another video. It's about gel filtration. Okay.
Separation in gel filtration chromatography is based on the differences in sizes from biomolecules as they pass through a column packed with a chromatographic medium, which is a gel.
You see those seeds?
Molecules larger than the pores in the gel are unable to diffuse into the gel and are confined to the solution surrounding the beads. Molecules which range in size between the very big, excluded from the gel, and the very small.
So these are the biggest molecule. They will not be trapped by this sieve, so they'll be excluded, so they will come out first, and then the medium size come out second, and then the smallest size molecule will come out last.
Can penetrate the pores to varying degrees based on their size. Access to the pores is limited by steric hindrance. If a molecule is smaller than the smallest of the pores in the gel, it will be able to enter the total pore volume.
So that's the gel filtration, which is a size exclusion chromatography. Okay. And section is about electrophoresis. The electrophoresis, basically, we try to separate the protein mixture, and then we can evaluate the protein quantity, quality, and also the protein size, right? So we run the electrophoresis, and the electrophoresis basically this is a typical setup for SDS page gel, all right? And then because the protein, they have different charge, different size, and different shape, and then they will have different mobility. So we can use these electrophoresis to evaluate the protein quality, quantity, and the size. And just to remind you that the aqueous gel normally is used for the nucleic acid separation. And the polyacromide gel, the SDS polyacromide gel, that's for protein, although sometimes we can use that to separate DNA as well. And for the nucleic acid separation, when we use the aqueous, because the nucleic acid is negative charge, and then we place the aqueous gel in the electric field and then connect the electricity from negative to positive. And so the nucleic acid will be able to migrate from negative to positive. And during this process, because the aqueous provide the complex, then they will restrict the migration of the nucleic acid. And so later on, during through the electrophoresis, so we can separate the small nucleic acid from the large nucleic acid, because the small nucleic acid can penetrate through the aqueous pore faster than the large molecule, so that we will be able to separate the DNA, RNA based on their size. And then for the protein. So as we learned from the previous chapter, protein, it depends on the amino acid, and that gives you different molecular weight. And also protein, the overall, the peptide, the protein, because different amino acid will provide different charge. It could be the non-polar, polar, positive charge, negative charge. All right. So it's not even. All right. So we have to process the protein before we perform the electrophoresis, so that we'll be able to separate the protein based on their size in the electric field. Okay. So that's a very important point, that you need to treat the protein sample before you run the electrophoresis. So one of the important detergent that we use is the SDS, which I will explain later. All right. So that so the idea is we want the small protein to move through the polyacromide network faster than the large one, so that we can separate these small protein from the large protein, and also the distance the protein travel would be inverse to the size of the molecule. Okay, so so basically we want the protein to be separated based on their size, all right, in the SDS page gel. And the reason we use the SDS is because, all right, so if we want to coat the protein, right, coat the protein evenly with the negative charge on the surface of the peptide. Therefore, we can make the protein molecule to migrate from the negative charge to the positive charge. All right, and then to coat the peptide with these SDS, the negative charge evenly, we have to first of all denature the protein. All right, the native conformation, we have to denature to make it linear. All right, to make it linear, then we have several different approach. All right, so the approach including the heat, it's not here, heat and ionic detergent and reducing agent to break the conformation. Right? And also the SDS, because we want to make sure the denatured protein can get a uniform charge to mass ratio. So you must disrupt the protein conformation. And the SDS normally is included in this laminate sample buffer that we normally use, all right? I have SDS, and then the bromophenol blue, that's a tracking dye, so that we can track the distance the protein migrate on the SDS page gel, because normally when you perform the gel electrophoresis, you don't know where is your sample. So that's why you need to add the tracking dye so that you will be able to see where is the protein, all right? So that's the function, that the purpose that we use the SDS. And then these two chemicals, the reducing agent, beta-mercaptoethanol and the DTT, they are important because they can break the disulfur bond, right? They can break the disulfur bond. And then the heat also can break those hydrogen bond. Is anyone having the same problem that my voice in and out? So you have difficulty. So when you answer yes, that means you have difficulty to hear me. That is clear. All right, so let me just try to fix this. Just a second. Let me see what I can do. Is this better now? Adele, you say yes, I mean it's better. All right, so it's better. Okay, so let's continue. So remember, I mentioned that SDS, all right, the ionic detergent, it's very important, all right? So the reason we use SDS is we try to coat the peptide protein evenly with the negative charge, all right? And then before we do that, we must denature the protein. So to denature the protein, you need heat to break those hydrogen bonds. Remember, where are those hydrogen bonds? The hydrogen bond in the secondary structure. The hydrogen bond is between, they are between the CO and the NH in those peptide bonds, right? In those peptide bonds, they form these hydrogen bond. And also in the tertiary structure, you want to break those hydrophobic interaction, which are those van der Waals interaction, and also the ionic interaction. So these are, and also the hydrogen bond between those polar amino acids. So the heat will help help us to break those interaction and make to denature protein, and plus the reducing agent to break the disulfur bond, so that we will have the completed denatured protein. All right, and then the SDS will be able to coat the protein evenly with the negative charge. All right, and the that's for the electrophoresis. All right, so that after the electrophoresis, in order to visualize the protein, right, we can use a different staining technique to visualize the protein on the SDS page gel. So here I listed like the Comasi brilliant blue, zinc reverse staining, and the silver staining. So the Comasi brilliant blue, basically you have two different modifications, right? It's an anionic triphenyl meson dye, right? That's an anionic triphenyl meson dye. So we have the Comasi R250, that's a red color. And then we have Comasi G250, that's green color. And so basically this dye will bind to the amino group of the protein, okay, in the presence of the acidic media. So basically, we normally add the acetic acid, all right, when we try to stain the gel. After the gel electrophoresis, we remove the SDS gel and then incubate in the presence of acetic acid, then you can add the composite either R250 or G50. Then those dye will bind to the amino group of the protein. And they bind the protein in a way that because it's a quantitative binding. So the more protein you have, then the more dye will bind to the protein. And then you will see even a heavy band. Yes, Yana.
Hi, is this chapter 5 still? Yes, right?
Yeah. It's still chapter 5, but it's not today's quiz.
Okay, yeah, because I can't find this slide on the PDF that you uploaded.
Oh, perhaps you should download one more time because I just updated the slide.
Okay, was this the only chapter that you updated the slides, or did you update the other chapters as well?
I only updated chapter 5, that's it.
Great, thank you.
You are welcome. Okay, so that's this dye will bind protein quantitatively. Therefore, the more protein you have, then the more deep color you will see. All right, and so it's a way that you can quantify the amount of protein. All right, and the zinc reverse staining is also called negative staining, and based on the precipitation of the zinc imodiazole on the gel. Okay, so that the zinc reverse staining can be applied on a composite blue stain gel, and then those undetected bands will appear in the zinc reverse staining. Okay, and silver staining, which is a very expensive staining because you have to use the silver to stain the gel, but it's very sensitive. Okay, it's very sensitive, and so most of time, people just use the composite brilliant blue to stain the gel. That's easy and cheap. So and also you can also cut the band, which represent a specific protein, and then for the downstream analysis, including the mass spec analysis. Okay? And this is just a typical example, the image of the composite blue. All right, the composite blue. It's the most frequently employed technique for protein detection in SDS gel, right? So you can see this example of the composite brilliant blue staining, and this is the zinc reverse staining, and this is just basically the same gel, and then you stain with the zinc reverse staining. Then you can see the band appear here, is not appear here, all right? And this is the silver staining. The silver staining, okay? It's very sensitive, but it's expensive. And also you can use the fluorescent staining technique to stain the gel, okay? Because these are the colorimetric staining method, and this is the fluorescent staining method. And that can give a very wide linear dynamic range over the 4 orders of the magnitude. These are lists of the fluorescence dye that you can use to stain the gel. Okay. So because the SDS page electrophoresis is so important, so I'm going to show you the video about the gel electrophoresis, because later on we will discuss the two-dimensional gel electrophoresis. It's all the same principle, but the one-dimensional SDS page gel, it will be the foundation. Okay, so we will watch this now.
Page or sodium dodecyl sulfate polyacrylamide gel electrophoresis is an electrophoresis method and very powerful technique in the study of proteins. In SDS page, proteins are separated in a polyacrylamide gel based on their molecular weight.
So just So you have SDS, so that will give the protein negative charge, but beta capital ethanol, or DDT, that's reducing agent, to break the disulfide bond. And then you have bromophenol blue, all right, to track the protein electrophoresis, and glycerol, just to make sure the sample will go down to the bottom of the well, so that the sample will not disperse in the liquid solution.
Samples may be biologically derived, for example, from prokaryotic eukaryotic cells, tissues, viruses, environmental samples, and purified proteins. When the sample buffer has been added, the samples are then heated at 95 degrees Celsius for 5 minutes.
So that's the heat, right? That's the heat to break the those weak interaction, like a hydrogen bond, hydrophobic interactions, ionic interaction, and also some hydrogen bond between the polar amino acid.
Proteins are large biomolecules consisting of one or more long chains of amino acid residues. Acids are organic compounds that contain amine and carboxyl functional groups, along with a side chain. Proteins are formed by linking amino acids with peptide bonds, and fold into one or more specific spatial conformations, driven by a number of interactions such as hydrogen bonds, hydrophobic interaction.
So these are between the polar amino acid, and then these are between the hydrophobic non-polar amino acid, and this is between the cysteine, right? Cysteine, the reducing agent, will break this disulfide ionic interaction.
And ionic bonds. SDS is an anionic surfactant and contain a polar head group with a net negative charge at the end of a long hydrophobic carbon chain. SDS denatures the native proteins by disturbing the hydrogen bonds, hydrophobic and ionic interactions. Is used to clean the sulfide bonds. When a protein mixture is heated, SDS binds uniformly to the protein, and then to the negative charges controlled by SDS. This treatment brings the finer proteins down to linear molecules with net negative charge. Therefore, these proteins can be separated in a polymer chain based on their molecular weight. The separating gel solution with pH approval and the standard gel solution with pH 6.8, and both consist of a polymer, this polymer, ammonium protothiophene. The gel is produced by polymerization between two waxed wafer and global films. The separating gel is not removed.
So the audio is not clear. And my voice is clear, right? Just not the video. Okay, so we will just go through this briefly. So basically, just tell you how they make gel, and then also those chemical and their function, right? To set gel, and then you set up the. So you set up this SDS page gel, and then you can see the gel on the top. You have the well, right? And then remember the negative, positive, and then you have the buffer, so that you can have a buffer to neutralize the change of the pH during the electrophoresis, and then you pour the gel, and then you load your sample, and the glycerol will help the sample to settle in the bottom of the well. All right. And then after you load the sample, and then you apply the electricity, and then the gel, the sample start migrating. All right. And during the migration, all right, the protein will penetrate through those polyachroma network, and then based on the size of the molecule, the smaller protein will penetrate through the polyachromate gel faster, and then the large one is slower, so that in the end, the distance of the migration will be different, because smaller molecule has the longest distance of the migration, and large molecule has a shorter distance of migration. All right, and that's how you separate this protein. All right, so in the end, you separate this protein. Okay, so you can separate this protein based on the size. Okay, and then after finish it, then you can stain the gel. All right, stain the gel, bromophenol blue in the presence of acetic acid. Then you will be able to visualize the protein. Okay, so this is the procedure of the standard procedure of the SDS page. Okay, and the kilodalton, that's the molecular weight of the protein. Okay. So that's basically the overview of the electrophoresis. Now, so the SDS page, it can give a very good resolution of polypeptide. But sometimes a mixture of polypeptide is very complex, that we need an even better method to resolve LNO. So and this technique to resolve the even more complex protein, it's called a proteomics, right? Proteomics. The proteomics can help us to separate and identify an even more complex protein mixture. Okay, and proteomic tool that can be a two different type of proteomic. One is a gel-based proteomic, right? Gel-based proteomic. So we have this two-dimensional gel electrophoresis, or you can have a gel-free proteomic that include the so that would be the liquid chromatography and or the capillary electrophoresis. So these are the useful methods to identify protein. Okay, and currently the proteomics has been dominated by this two-dimensional gel electrophoresis coupled with the mass spec, right? And it's very powerful technique to evaluate and analyze the protein. So for the two-dimensional gel electrophoresis, as we know, that one of the dimensions would be this regular, the standard protein gel electrophoresis. That's SDS page gel. That's based on the size. Okay, based on the size. For the for another dimension, that would be isoelectric focusing based on different isoelectric point of the protein, so that we will separate the protein based on isoelectric focusing, the PI, and then also separate the protein based on the size, so that we can distinguish every single protein in the mixture. Okay, and so basically this is a typical example of a two-dimensional electrophoresis. So first dimension, first of all, you have to separate the protein, right, based on the isoelectric focusing in one direction. Okay, so in one direction, right? So this is, you have increasing pH, all right, and then you run the protein mixture, and then those proteins will separate from each other based on their PI, all right. And based on different PI, then you can see all these different proteins line up on this first dimension. And then, so these different pH, so for example, pH 5, pH 6, the pH 5 could have 20 peptide here. pH 6 could have 100 protein here. Then you have to cut this gel and then put it horizontally on this SDS page gel. Okay. Then you run the second dimension based on the size, like the previous standard SDS page gel. All right, so that the same PI, then they can be separated based on the size. All right, so for example, these band here, all those proteins, they have the same PI. However, when you run the second dimension, then you can see 3 proteins here because they all have different. These 3 proteins, although they have the same PI, but they have a different molecular weight. All right, so that's how you separate the complex protein mixture. And these are the typical result of a 2-dimensional gel, right? Once you have the result, then you can cut the protein at a specific location and then perform the mass spec to analyze the protein, okay? So this is dimensional gelophoresis. Any question? Very good, Hadir. Where's the other half of the class? Ido, are you here? Ido. Are you here? All right, so that's the protein gelophoresis, okay? And then the next technique is to determine the primary structure of protein. Okay, so basically the primary structure of a protein, that's basically amino acid sequence. So that you want to determine which amino acid are present in the peptide. Okay, and then you want to determine the N-terminal and C-terminal of the sequence. And you want to determine the sequence of smaller peptide fragment. Okay, so these are very important. So basically, these are, let's say you have the peptide. Then, first of all, you want to hydrolyze to the constituent amino acid, and then so you can identify individual amino acid by HPLC to see which amino acid present in the peptide, and then you use the special reagent to determine the N-terminal, C-terminal, and then you cleave the sample with different chemical so that you can determine the sequence of you cut it into the smaller peptide, and then you can determine the sequence of the smaller peptide. Okay? So that's how you determine the primary structure. And so this is the amino acid analysis, AAA, so that you can see the presence of the individual amino acid in the and the protein cleavage, all right, protein cleavage. And here the protein cleavage, all right, steps through here. So we can use different enzyme and chemical reagent, okay. So for example, the enzyme, the trypsin and that chymotrypsin that we can use, because the trypsin basically will cut the C terminal of the positive charged amino acid. So for example, they will cut the C terminal of the histidine, lysine, and arginine, that trypsin. And the chymotrypsin will cleave the C terminal of aromatic amino acid. So which amino acid has aromatic ring? So we have tyrosine, phenylalanine, and the tryptophan. Okay, so these three amino acids are the aromatic amino acids. So the chymotrypsin will cut the C terminal of the tyrosine, phenylalanine, and tryptophan. And trypsin will cut at the C terminal of the histidine, lysine, and arginine. And then the chemical reagent, the cyanogen bromide, will cut at the junction where you find the methionine. Okay. So by using these different enzyme and the chemical reagent, then we can degrade this long piece of the peptide and make it into the smaller pieces, and then you can determine their sequence. All right. So here, for example, you apply the trypsin, right? Trypsin will cut those positive charged amino acid, so that when you apply the trypsin, then you will cut here. See the arginine at C terminal, arginine and lysine, all right, C terminal here. And then you can produce different smaller peptides. So if you treat this peptide with trypsin, then you cut here arginine, right? Cut here lysine, arginine, so that you produce these 5 different peptides, smaller peptides. And then you can also use the chymotrypsin, right? Chymotrypsin cut the same protein, and then that will cut at the location where you find the aromatic amino acid. So that tyrosine, tryptophan, phenylalanine. So here, see, we'll cut here, here, here. All right, and so you obtain this smaller fragment, smaller peptide. If you can combine the digestion, then you will be able to interpret the peptide sequence. Okay, and this is the cyanide bromide to cut the internal methionine. All right, to cut the internal methionine. So that here's the example to determine the protein sequence. So basically, if you treat the peptide with the chymotrypsin, then you cut it the phenylalanine, and the cyanogen bromide, you get the methionine, and then chymotrypsin, and then you get another fragment. Is this fragment, and then cyanogen bromide, another fragment, another peptide, and then so if you use these different reagent and enzyme, you get these six different peptide. Then by doing those arrangement, then you can figure out the correct arrangement of the peptide. All right. Question. This will be the exam. Be on the exam 2. All right. So to analyze the peptide, and then through different digestion, then you'll be able to arrange those small peptide, and then to come up with these.
Professor, can you give us another example, please?
The example is here. So you use of the overlapping sequence that to determine the protein sequence. So basically, it just depends on what the enzyme and yes, there, yes, different enzyme, different chemical reagent, and then from those small peptides, then you will be able to try to interpret it, all right, to arrange those small peptides.
So on the exam, we have to determine the protein sequence, or how,
yes,
how exactly would you ask?
Yes. In the exam, for example, in the exam, you will not see this overall sequence, all right? In the exam, I will give you, okay, the chymotrypsin give you how many different fragments, different peptide, and what's the sequence of those peptides. And then if I use a trypsin, what kind of peptide you get, and then you will try to look at those different smaller peptides, and then to make the arrangement to come up this answer, like the bottom one here. Okay, Hadel.
But you said yesterday 1 to 5. This is not 5.
What did you say?
You said yesterday 1, 5, 5 plus 1, 4. This is past that part. So we didn't really prepare for this part.
No, this part is not on the quiz.
You just said it was on the quiz.
No, you'll be in the next quiz, next exam.
Oh, okay.
Yeah, next exam.
Thank you. Thank you so much. Sorry about that.
No problem. Whatever that we taught from Monday and Tuesday will be on today's quiz. And whatever we're going to go through today will not be on today's quiz, okay? And especially like these will be on the exam, yes. So that's peptide sequence that you use to do the to determine the sequence. And this is the sequence in the peptide by the Edman method. So Edman method basically, so in the first step here, all right, the first step here, you use the this phenyl isocyanide, okay, combine these with the N-terminus of the peptide under the mild alkaline condition, so that they can form this phenylcyl carbonyl substitution. All right, from this substitution, and then the second step, you treat this complex with the trifluoroacetic acid TFA, and then this cyclize to release the N-terminal amino acid residue as a silazolidinol derivative, right? And then, but the other is not hydrolyzed. And then you continue. Then you can just so the organic extraction and treatment with the aqueous acid that will yield the N-terminal amino acid as the phenylcyl hydantoin. That's a PTH derivative. And this process, you can repeat this process with the remainder of the peptide chain, and then you can determine the N-terminal amino acid one at a time. Okay, let's go Edman. And then to identify the protein, all right? Identify protein, you use a mass spec, right? You can use a mass spec to determine the protein. Yeah. And this is just the example the multi-top mass spec. So you have a protein sample, you place on this surface, and then you have laser beam to excite the protein so that to give this protein positive charge, and based on the molecular weight, and then they carry different charge of protein. And then they will go through this the gap, this focusing length, and then they will go through this gap of one by one, and then they go through that, and then you have to detect each of the protein molecule, and then they can determine the protein sequence. That's the Martov mass spec. So in the chemistry department, they perform this kind of analysis to determine the protein sequence. And then this is another way that you can identify the protein through this ELISA. Okay, so that you have these. Okay, so this is another technique. You can identify protein. So for example, here you have the protein, right? And then the primary antibody will bind the recognized protein and bind it. And then you have the secondary protein to recognize your primary protein, but the secondary protein carry the some enzyme, all right, carry some enzyme or some marker, all right, some marker, and then this marker will give you the signal after you add some the if the enzyme catalyze a specific reaction, and then you'll be able to determine the presence of a specific protein. Okay, so that's another technique that you can use. And also Western blot is a very powerful and commonly used technique to identify the presence of a specific protein. So for the Western blot, okay, so basically the first step is to run the protein SDS page gel like a regular protein gel, and then after the electrophoresis, then you transfer the protein, all right, from the polyacromide gel to a membrane, so like a nitrocellular membrane or nylon membrane, and then after you transfer those protein to the membrane, then you can so this is the standard procedure for the electrophoresis, and then you the filter paper gel and then nitrocellular membrane, and you transfer the protein from the gel to the membrane. Right after you transfer to the membrane, then you incubate the membrane, all right, with the antibody, and then this antibody is a specific antibody that you want to use to find your target protein, and then if the target protein is present on the gel, then you will see the signal, all right. So like this result here. So after the SDS page gel, you transfer the protein to the membrane, then you use the primary antibody, and primary antibody will bind to recognize those protein, your target protein, and then you have the secondary labeled secondary antibody, because the secondary antibody will help you to visualize the location of the protein, and then you will detect the label. So on the left here, now will be the protein gel, SDS page gel, and then on the right, now will be the Western blot. Now you take the gel, and then you transfer all these protein to the membrane, and then you incubate with the primary antibody and secondary antibody, and finally you see the band here. So that just proves the presence of the specific protein. So this is Western blot. And then another technique you can use is called protein chips, which is similar idea for the ELISA experiment, okay? But instead of one sample at a time, then you can perform 1,000 samples at one time, okay? So that's protein chips. So that's basically the end of the chapter 5, okay?